experimentation is a subject which provokes passionate arguments between defenders of animal rights and those who consider laboratory animals as a necessary evil for the advance of science. It's a subject which touches the general public. Animal lovers, people that share their lives with animals, find animal experimentation traumatic.
The principal objective of animal experimentation, at least in the public mind, is biomedical research, and therefore everything which involves sickness and death. And so there is a kind of self-projection into sickness and into death, which leads to rejection of this practice. The other aspect,
within minutes,
there
will be a significant drop in the contaminant levels in this area.
Noble and specifically human ideas, that is the advancement of knowledge, and the fact that in the framework of that advance, an animal will be made to suffer.
I will be breathing in nanoparticles as I stand here. When I go back in the car,
of emotional values
our
for
roads are
animals
full of
used in
nanoparticles
laboratories.
from exhaust.
We can
What
see
is
that the
new
public is quite
the
insensitive to
deliberate
rodents,
engineering of
quite
these very,
indifferent to
very
the
small
pig,
particles
which is an
to
extremely
have
affectionate animal,
enhanced or
and that
even
the
novel
public is
properties.
devastated by the use of dogs,
And the
cats, and
uncertainties that
primates.
that introduces in terms of the potential risks to the environment and health.
In reality, interest for animals is driven by an anthropocentric vision, which means that it is centered on mankind. Humans have a tendency to only be interested in animals which are close to us or which resemble us, in such a way that the ethical attention of man to animals is established in concentric circles, a kind of downwards gradient, which means that the further away an animal is from us in its shape, its behavior, the less interested in it we are. So, close to us we find obviously household pets, dogs, cats, and next comes horses or the great apes, then farmyard animals, and then birds, next reptiles, followed by fish, and after that, well, then there's the vast, immense population, which is around 95% of the living world, the invertebrates, the insects, which nobody really cares much for, except for specialists.
Animal experimentation is a social question which forces us to reflect on our relationship with other living creatures and on the hierarchy of the living world which we establish. For centuries, the animal world and humanity have been kept carefully separate. Human beings were far and away the most intelligent and sensitive creatures, and refused any possibility that the beasts could have intelligence, sensitivity, or emotions. However, through their experiments, scientists are proving little by little that the frontier between the two worlds is not as uncrossable as was thought. Just like us, animals learn, memorize, imitate, communicate, feel, and dream. From this point on, our attitude to animals and the way in which we treat them can only change. Discussion about the possibility of replacing, reducing, and rationalizing the use of laboratory animals was begun at the end of the 1950s. This was the theory of the 3 R's, developed by 2 English researchers, William Russell and Rex Birch, who formalized their ideas
There's 2
on animal
aspects
experimentation,
to these solar
working from
technologies that are
ethical
incredibly
and
exciting.
scientific points
The
of
first
view.
is just raw performance to cost. The other thing about having a flexible solar cell or a paintable solar cell is that it just allows your imagination to go wild.
Replace animal
Imagine if you
experimentation by using other
could
methods,
wear
to
clothes
reduce the
that were
number
solar
of
energy
animals
harvesters
used,
and
and to
would charge up
rationalize
the batteries
experimental
of the
protocols
mobile
to
devices
limit
that
the
you were
suffering
carrying around.
inflicted upon
Imagine
animals.
if you could create a sailboat whose sail was also a solar energy harvester and can charge up the battery on your sailboat. The imaginative possibilities that emerge when you start to make low-cost, flexible, and high-efficiency
The
solar
question of
cells
suffering is
simultaneously
central as, for
are
public
really
opinion,
limitless.
animal experimentation is above all another way of saying trauma and pain. if attention was mostly centered on mammals, the ethical question of suffering exists for all living creatures, even for those that mankind considers to be less evolved.
After having demonstrated that some invertebrate animals could feel pain, we managed to get the new European directive to take cephalopods into consideration, for the European community to recognize them as sensitive beings and which are now protected like vertebrates are.
Anesthesia is the most common method of limiting the suffering of animals during experiments.
I'm quite shocked by the reactions to this subject that can be heard
Solar
about
to us is very
scientists who are
important.
sometimes accused
Basically,
of
we had
being
the challenge from
sadists,
the chairman of
heartless
Cow's board
people who torture
telling us
animals.
that he wants
No,
Saudi Arabia in the
it's
future
untrue.
to export
For one
the same
simple
BTUs
reason,
that we are
and
exporting
that is
from
that a
fossil
mistreated
fuel
animal
in the form
endangers
of
the
solar
validity of the
energy.
experiment.
So
if
I don't
only
know
for
how
that.
big that challenge is, but it's
But there are
really
other reasons.
a tall order challenge.
Researchers are attentive to the well-being of the animal. Furthermore, they do all they can, everything that is possible, so that the animal is damaged as little as possible during the
It's a bit like
experiment
when,
they make upon it.
I guess it would have been in the 1950s, people wondered whether one could go to the moon. And it became clear that there's no physical impediment. There's no laws of physics that would prevent that from happening. And it became a technology challenge.
Despite the adoption of the 3 R's rule by the scientific community, science still uses a lot of animals. 12 million a year for the 27 countries
Energy
of the
today
European
is a
Union,
$9 trillion
of which more than 80%
industry.
are rodents.
I can't think of a bigger sector of the economy. I mean, it's absolutely huge. It's something
However,
we almost
the
don't notice because
reliability of
it's so
animal
much around
experimentation
us
can
and we
be
take it for
questioned.
granted.
Are results of tests on animals capable of being applied to man?
Does the eel show you like the elemental makeup of glycogen? Or?
In order to limit the risks of error in such extrapolation, some research is trying to rely rather on direct observation of human subject. This is the main aim of a new study in respiratory toxicology being developed by
The
Professor
sun's
Gretschert.
power reaching the Earth is really incredible. There's 10,000 times
By our
more
research
solar power
on
reaching the
exoplates,
Earth
we
every
do
day than we use
observations
across
in
all our
humans
energy
to see what happens
habits.
in the
Another way to
airways,
put the same
and we
idea is
especially
that
focus on
as much sun
children
hits the
because they
Earth
are a
in an hour
vulnerable population and
as we
they
consume
suffer
in a
from
year.
respiratory diseases. 1 out of 3 has asthma or allergies, and by looking at the exhale breath, we try to see what happens in the airways.
One of the phenomena at the heart of
Once when we have the blood
nanotechnology
samples
is
of
that
the
we can
children,
shape matter.
we
As a
analyze them,
consequence, in
and
my
we
lab,
get a spectrum
when we
which,
make
where
3
you
different
can
batches
see
of
the different
nanoparticles, we can
molecules which are
program them,
present, and
each
we
to be
can
responsive
identify
to
which
different
chemicals
slices
are
of the sun
present here.
spectrum.
It's on the chemical molecules identified in the breath of children that Professor Gretschert will concentrate her research upon by developing a very specific method.
We culture airway cells which come from human airways.
And so
And we
it turns out that
culture
fully
them, and
half
then
of the
we
sun's
expose
energy
them to
lies in the
chemicals which are
infrared
present in the
frequencies
environment.
and the other
we can
half
see
in the
which
visible.
effects the chemicals have
So
on these cells.
my research
and this we can compare
acknowledges
with what
that
you observe
we need to
in
capture
the children.
all of that energy if we're going to make an efficient solar cell.
With this equipment, we simulate exposure of the human lungs to air with chemicals. And what we do is we
So the
put
major
cells in
differences
these
in
little
making
wells,
quantum
and
dot-based
they
solar
get
cells
air with
relative
chemicals
to
on
existing
top
approaches.
of it.
The
And
first is that
underneath
we
there
actually do
there is
work
some
with
liquid,
individual
and that
nanometer
simulates
sized
the body
crystals, and they're
fluids.
individually packaged. So each one is in a position such that we can just coat it,
For
spray it, if you
respiratory
like, paint
toxicology,
it
it's
onto a
especially
vacuum.
important to work on human cells because there is no good animal model available. The metabolism in an animal is different from in humans for many chemicals, and also animals
and then also
don't
by
live
virtue
that
of their
long.
lower cost,
So while
you won't
humans
hesitate
can
to
be
deploy them
exposed
across
for
the
many
entire
years
surface
to a
of the
chemical,
building.
it's
In
very
fact,
difficult
if
to
they're
simulate
cheap
that
enough, if there's a little bit of power
by
to be
animal
had from the
tests.
sides of the building,
So
you might
that
even put them there
complicates
too.
the extrapolation from animals to humans.
Today, certain cell culture techniques allow the complete replacement of animal testing. In total, 30 or so alternative methods are used in Europe, of which 2/3 concern cosmetics.
I've been living in this city all my life.
The search for alternative
Over the
methods
years,
of testing cosmetic products
I've seen changes
has also been
I
stimulated
never
by
would have
more
believed.
restrictive legislation.
Take power.
Animal testing was
My
banned
grandson
in
just
2004
assumes it
for
comes
finished
from the sun.
products.
But
Since
it wasn't always
2009,
like that.
legislation applies to all the
There used to
ingredients
be
used
coal
in their
and
composition.
oil. And who could forget all the trouble with nuclear? After that, energy saving came back.
For the last 20
People
years,
bought all
oil
kinds of
research
do-it-yourself
laboratories have been
solar
working
stuff,
towards
like
making human
my mini
corneas
panel.
or skin tissue from
It's
cell
basic,
cultures.
but I'm still using it. Then came the Nanoflex fabric balloons. At the beginning, they were kind
These
of
tissues
modest.
made in laboratories and
My
now
neighbor
used
Sam was
instead
the
of
first
animals to
person I
test
know
for
to
dermal
try one.
irritation,
Pushy guy.
corrosion, and phototoxicity.
Always wants to be first with everything. Yeah, did seem like
We
a bright
can thus
idea.
measure the effects of a product in different forms. Liquid,
Right away,
cream, paste,
there were
or
tensions.
powder.
And in no time at all, there was trouble.
Sam! It's that damn balloon of yours! Keeps blocking the sun from my solar panels. Now there's no hot water.
Hey, sunlight is first come, first serve. Go get yourself your own balloon. Hey,
You can't hog our sunlight.
hey, I can't help it. If my balloon happens to be bigger than your panel.
I'm gonna tear your damn balloon to pieces. Shut up, you solar thief!
Nous avons devant nous
So we have before us a panel of different kinds of artificial skin tissues,
Sam's
ranging from the
Nanoflex
least to the most
balloons were just the
sophisticated.
beginning.
We've got here what is called total
People
artificial
started to
skin
think big.
with living epidermis and living
Do I mean
dermis.
big?
2 cell types, and if another cell type
Why
is
stop
added,
at balloons and solar
melanocyte,
panels when
which
you
will
can
give us
have your
melanin,
very own
skin pigment. Then we
nano-solar
have here skin
reflector
which can get a
airship?
suntan.
Solar guzzlers. That's what they are. Swallowing up the sky.
c'est quand même
It's still
incroyable.
quite incredible nowadays, and yet it isn't the first time we've ever seen this kind of skin grown from a few cells in a culture, and in a fortnight. We get something which looks like skin, so let's see. I'm taking this reconstituted skin apart. On this surface we have what represents the epidermis.
Sur cette surface, nous avons ce qui
That's the surface of the skin in contact with the air.
la peau qui est au contact de l'air.
And if we turn it over, there's this shinier surface which
We're
corresponds
in
to the
danger.
dermis,
We're in danger
which
because
gives skin its
we
resistance. And what we
need
see
water.
here
And the only source we have
is that
it's
this skin
poisonous
resistance
with
is both
poison.
what we find in real life and enables us to apply cosmetic products.
Ça nous aide pour
For example, lipstick or pasty substances that are difficult to apply.
This is the city
Well,
where I
the
was
surface
born.
has to be strong
And
enough
right
to
now,
have the
my
substances put on
responsibility
it,
is
then
to make
spread
sure
out to
that
imitate
everyone
real-life
at their
situations.
homes have water every time they need it.
Donc après avoir
So, after having frozen this skin at minus 180 degrees centigrade, we'll cut them. This is a slicer, like a ham slicer such as we have at home, except that these are slices only a few microns thick, so we can observe skin changes microscopically after they've been in contact with the product.
We are having water from 700 meters deep wells, and the concentrations of arsenic at that levels are very, very high.
The water coming out from this water treatment plant, it's pretty clean, but we still have a problem with the arsenic. And we cannot face it with the available technology.
We can see here the
because
different
it's too
structures of
expensive for
artificial
us.
skin, starting from the most external part, the layer of hard skin, the epidermis, and the deepest part, which is the dermis. So with this kind of cut, we can observe the potential effect of products applied to their surface. After treatment by an irritating substance, we can clearly see the disappearance of the hard skin and the appearance of white spaces which correspond to dead cells, and then the destruction of cell layers. Working with artificial human skin gives us more reliable results because, to start with, it's human cells, and further, a lot more refined and directly applicable information is provided with regard to what happens to people. Putting an alternative method in place in Europe could take between 3 and 10 years.
We can see those materials using our very latest instruments. We can actually know a lot about how small they are because we can go down to those length scales. But more than that, they're strange. Materials on the nanoscale, they interact with material, with things around them very differently. And they don't behave at all like things that are just a little bit bigger. So that strangeness is really the value of nanomaterials. When you find an application like we have here in Mexico, where that very strange chemistry can be coupled with a really important social need, that's when you have this opportunity to actually change people's lives.
This validation period is a crucial debating point which has been revived by the launch by Europe in 2007 of the REACH Toxicology Protection Program. This obliges manufacturers to prove the products they market are non-toxic. Between today and 2025, 30,000 substances will be tested. With this delay, can we manage to validate enough alternative methods to substitute for animal tests?
The REACH program was from the start hypocritical, as it had admitted that it would mean a considerable increase in animal experiments, but stated that animals couldn't be used if alternative methods existed. In the full knowledge that such alternative methods didn't exist, and as a result, it was thought that there would be 30 million more animal experiments in 10 years. Actually, it's more like 10 million, but that's still enormous.
To measure the effects of chemical products, it must first be known which
Well, I
organs
think everybody
they will
knows what
affect.
rust is. We're using a particular form of
Animals
rust,
are
and
used to follow the
nanorust
route
is a good
taken
way
of
to
a
think about it,
substance
but
in a
it's
complete
special.
living
It's special
organism,
because it's
which
really,
is where
really small.
cellular tests reach their
And so really
limits.
what we're hoping to do is by using these nanomaterials that have just in a single
In fact,
small
a
amount
cell
tons and
isn't an
tons and tons and
isolated
tons of
island.
surface area
It
for the
belongs to a
arsenic to
tissue
stick
which
to.
is part of an organ,
It's kind of like an
linked
amazing
to other
sponge for
organs
arsenic.
to form a complete organism.
Good. Well, want to do it right.
However, before attaching itself
Water
to an
everywhere
organ,
is getting
a
scarcer, it's
supposedly
getting
harmful
harder
molecule
to
will
clean
pass
it.
through
We're having
different
to dig
organs and
deeper
cell
to find
structures
aquifers
and
that
can
are
cause
not already
collateral
been used by people.
which
And
is
in fact,
impossible
arsenic
to see
becomes
by
more of a
testing in
problem.
vitro.
Like in this community, is they have to turn to deeper and deeper wells. Those always have more and more arsenic. So it's an existing problem, and it's certainly one that we anticipate growing,
However,
not just
cellular
here, but in
tests
many,
have
many other
considerably
regions throughout
contributed
the
to the
world.
way we can estimate the harmful effects of
Hey, guys.
products
Hey,
and how
Vicky.
they act upon us.
How's it going?
It's going.
It's like a
Ça fait
column.
un siècle
Oui, bueno.
que
We're hoping to
l'industriel
put
voit la
10,000 liters through that 1 pound of
Manufacturers
magnetite.
saw predictive toxicology in terms of what is called lethal dose 50,
ils ont un produit et ils vont
which
regarder
means a product
quelle
is
est la
taken
dose
and it is seen what dose administered to animals, eventually the dose used on cells, that will cause death
Give me a good
in
twist.
50% of the animal or cell sample.
Yeah, all right.
You guys ready to clean some water?
En gros, on
Yeah.
To say
All right, it's time to
crudely,
take you to the field.
that toxic doses were aimed for.
l'idée qui prévaut
There's been a total change, and the prevailing idea now is that well before trying to destroy cells or animals,
un produit quel qu'il soit.
whatever product it is, will modify cellular molecular circuits.
We're pumping the
And
groundwater
this, in
directly
such
into
a
the top
way
of our column
that
with
repeated
arsenic in it.
doses
L'augmentation
or increased doses could effectively lead to lethal toxicity
Nanotechnology is this capability, this new, entirely new toolkit that people have available to them. And I think it's going to move into just about every arena, limited perhaps only by the creativity of people and how they take these new ideas and new tools and solve some of the world's most hardest problems.
To be able to work on these channels, it is essential to have laboratory access to human cells with human molecular mechanisms, from the main organs which toxicity will affect, liver, heart, kidneys, brain, and also skin and muscles. So stem cells are becoming a tool, a dream tool for this kind of thing, that can be produced in the requisite quantity and made to specialize in the tissue.
Well, on the other side of the building here, they would have had a maintenance yard where they would have cleaned up tools and trucks with a trichloroethylene, which is a chlorinated solvent that's commonly used for degreasing. It's also commonly used in dry cleaners. Historically, let's say 20, 30 years ago, when you were done with oils and these kind of liquids, you just pour them on the ground or pour them down a drain, and they let it get into the substrate. So we didn't really understand that these contaminants would persist for long periods of time, that they're extremely toxic.
We humans, you're not just looking at cardiac tissue or hepatic tissue, but an ensemble of integrated and interlaced systems. And at some point, we need to know
for about
all of
3.5 hours. As long as the water is moving, it's not going to freeze.
A lot of ground by looking in a laboratory.
Watching for this to turn orange, then it's going to be ready to dissolve.
A lot closer to human reality because they are human cells, and because with human cells we can reproduce organs which are otherwise mostly inaccessible.
Des organes qui sont quasi inaccessibles. Autrement.
What do you want to add? Turn up the red one. Yeah, it's frozen.
It's frozen.
Nowadays,
these conditions do present significant challenges with the freezing lines, the generators, the compressors harder to start. Having said that, though, the contamination is moving over this way here. So the longer you wait, if you wait 6 months, it's going to be that much further down, contaminated more area. So we want to get at it as soon as possible.
Unless we go to many patients, and that's what is done nowadays when a medicine is being tested. Hundreds, even thousands of patients are tested to test genetic diversity.
So until we get it into the vessel, which is in an exclusion zone, we have to be very careful. That's why we have to kind of stay back.
Where the toxicity
this is the exciting time. We're going to inject the borohydride into the synthesis vessel. It's going to turn black. And if it doesn't turn black, you're going to have some worried people.
What we are not able to examine with stem cells is systemic biology. That is to say, the integration of different organs and life, and in reality,
it's not yellowish anymore or orange in certain plants and nanoparticles now.
Cannot be replaced. What will always be extraordinary
420 mils in how many seconds?
At any moment it can
8
produce
seconds.
responses
That's crazy,
which were precisely what was not expected, and it's that aspect that's important.
So we're injecting the nanometals right in here, and what I'm doing is I'm keeping a close eye on the water level because we want to make sure it doesn't come up and spill over the wellhead here. It's going to go in there and it's going to go towards this well here. Just stay there in case it comes up. It'll be like a gusher.
Chosen at random. We study particular species of animals, those which are as close as possible to what we think are human ways of functioning, to be able to transpose results to humans as reliably as possible. We can consider that results obtained from
we're going to have a smear zone around this entire zone to attack the
humans.
contamination in this area. The nanometals that we're injecting today, we inject them in for two purposes. One, to attack the initial contamination in this zone, but then there's going to be some residual nanometals in this area. So as the water continues to flow through here that's contaminated, it'll flow through this residual nanoparticles and they'll be degraded. So further on downstream, the water will be cleaned and you can probably drink it.
We are still far from being able to artificially create the complexity of exchanges in an organism, but progress in cell cultures allows us to begin to simulate biological ensembles
stop,
by linking
and you see
the cells
a couple of
some
intersections that are
organs in networks.
empty with a couple of wells sticking out. It's like an old gas station that's contaminated where no one wants to go back in there and buy up and redevelop the land because it is contaminated
we can
at
artificially
significant
create
cost.
some kinds of relationships. But even there are limits. We can study exchanges in this way, but frequently a living organism has to be used.
Well, these nanoparticles are very reactive, so once we inject them into the ground, they don't last very long. They'll react with the contaminants in the ground, they'll react with the water, with air, and then they'll essentially become rust, and they'll aggregate together, stick together, and then they'll settle out a solution and dissolve away, so there'll be no problem.
While methods that allow us to completely replace animal experimentation are still rare, there are, however, several other possibilities that lead to limiting the use of animals. This is especially the case of medical imagery techniques.
The Nobel Prize was awarded for the development of DDT with the possibility of eradicating malaria. The use of asbestos as a fire retardant had tremendous benefits, but of course with significant impacts on human health. What we're trying to do with nanotechnology is avoid those sort of disappointments, to avoid those sort of environmental impacts.
It's okay, the rat's anaesthetized.
Yes, he's asleep, it's okay.
We have on our imaging platform studies which consist in studying the mechanisms of illnesses, following their progression over weeks in the same animal, so using fewer animals, and we're also studying the action mechanisms of new medicines. These medicines could be brain medicines,
what we're doing here is testing
psychiatric
nanomaterials
purposes,
in a very
and also
complex
could be medicines
environment.
targeted on the cardiovascular system, the pulmonary system, etc. So there, we can clearly see the MRI image of the rat's
As we
brain.
use these products,
And
these
so we
things
can
will go down the
completely
drain,
survey
end up in a
the region
wastewater
that
treatment
interests
plant.
us,
The
which
wastewater
allows us
treatment
to follow
plant will separate
changes in
them
this
into
region
the
during
clarified
the
water
rat's
and
sickness. So this
the
rat,
sludge or
we'll
the,
see it again next
the solids
week
that
to
come out of that
resurvey
process.
this region and thus evaluate the treatment over 3 weeks. It is estimated that imaging techniques in vivo with small animals use 5 to 10 times less animals
One of
than
the
the
things
classic
that we're interested in
techniques
looking
with
at
an
here is
invasive
what
approach
happens when we do
in
apply
tissue
them to land.
sampling.
How long will they stay there? Where will they
Imaging
go?
avoids having
What
to
kinds of
autopsy a
organisms
different
will they
animal
impact
at
along the
each
way?
step of an experiment.
On our platform, we have different imaging techniques, the MRI technique, but also others which are called radioactive techniques. Each technique answers questions and can complete one another.
When you talk
One
about a
technique gives
nanoparticle,
more
it's all
anatomical
surface.
information about the organ
When you get down
structure,
to a
the other
particle
gives more
that's
answers about
something
biological
like
changes in the
5
organ.
nanometers in diameter, about half of the atoms are on the surface. Nature will modify these surfaces just
The
as
image
much
shows
as
clearly
people
the distribution
will
of
modify the
radioactive
surface. So
tracer
we can
within the
engineer the
brain,
surface to
and
death,
also
but when
peripheral
we put
points.
it out in nature, nature
The
will begin to
important thing is
modify
our
that
medicine
surface.
gets
And we
into the
believe that
brain.
those modifications change the very properties of nanomaterials.
The interest of a platform such as ours is that we can make imaging examinations, whether it be on small animals like mice and rats, for example, or on more evolved models, larger ones, such as on primates,
So we're working with
or pigs.
killifish larvae,
and finally,
and
we're able to examine
dosing
human
them with
volunteer subjects,
one of our silver
patients
nanoparticles,
or
and trying
ordinary
to understand
people, and
how
so we can
different
get a
types of
continuum
water affect the
between the animal
toxicity of
model
those
and the
particles.
sick human at the end of the chain.
Another possibility in the development of alternative
Over the past several
methods
years, we've made a lot of
consists
progress in
replacing one
understanding
animal
how a
by
particle might affect an individual species, fish, invertebrates, etc., but understanding how it may alter the entire ecosystem or microbial nutrient processing, etc. We're
because in
just at
reality
the
there's
cusp
no real
of that type of
objectivity in this
work.
domain, and anyway it's not easy to be objective, is considered as an advance by legislators and the majority of public opinion.
We don't normally regulate most materials before they're released in commercial products in the United States. So generally we're left with a situation where we're trying to understand whether products that are already out on the market might pose a hazard to either wildlife or humans specifically.
Which have mechanisms
so we have to look very fast to try to understand these questions as quickly as we can.
Organisms, and
the
therefore
last
they
thing
can
anyone
replace
wants
a
is
lot
30
of
years from
mammalian
now
studies.
figuring out that there was a problem that we should have discovered earlier in the process. And so it is definitely a race.
You know what? Some things
We
are
have
never
breeding
quite
fish
what
in
they
the
seem.
tank, and now
This
in the
park, for
morning we
instance.
collect the eggs to start toxicity studies.
I live nearby and come here every day when my grandson's around. He'd play here all day if he could. This park was a wasteland when I first bought my little apartment.
The zebrafish
That was the only reason I could
development
afford the place.
can be followed within a few
Nobody wanted to
days,
live on
and
the edge
therefore the
of a dead
zebrafish
zone.
embryo does allow a fast screening
It was all fenced up
of
and no one
toxicology
could go
of
near it.
chemicals and pharmaceutical
But I was
drugs.
lucky. It was chosen for the latest nano remediation soon after I moved in. The whole area was off limits for years. No one knew exactly what they were up to, but in the end,
embryo at different
it was
developmental
amazing.
stages.
Magical
And here we have
to see a
normal
dark,
larvae
derelict
swimming in the
space
water,
come alive
while
again.
compared to the
A rundown
abnormal
wasteland,
larvae, which has been
transformed
exposed
into
to
a
a
beautiful
chemical,
park.
and where we see an edema
It's a joy
surrounding
to
the
see
heart
my grandson
and a tail
play here with
which
his
is
friends and
deformed.
think back on what it used to be like. But lately I'm not so
In the lab,
sure.
we have specific equipment to track
Protective
the
tents went up
movements of
again.
zebrafish embryo and larvae.
And the inspectors
by
were back,
tracking the
looking for
movements
something.
during 5
I don't know
minutes of larvae or embryos, we get a
Then
view on the potential
the anti-nano
toxic
troublemakers
effects of
started.
chemicals. This is a
Looks
track
like fear-mongering
of
to me, but
larvae
fear
which
can
is
be catching.
normal developed and which
I
is
don't know
moving
what
a
to
lot,
think,
as is shown
as I
by
say.
a lot of red lines.
These days,
compared to a
nothing's
larvae which
quite
is exposed
what
to
it
a
seems.
chemical drug and where you have less lines in the track. The work with zebrafish embryos is considered as an alternative method because the embryos, until the self-feeding stage, are by
The sources of
legislation not
uncertainty
considered as
in
animals.
this area are numerous.
And
We
this
don't know
allows
what
us to
nanomaterials
use
will be made.
zebrafish embryos
We don't know how they will be used.
similar
We don't know
to
how many
cell
will be
culture
used.
studies,
We don't
for
know where they
instance,
will go. We
as
don't know who
maybe
they'll
a
impact.
replacement to animal
There are
experiments.
a great number of unknowns. Some of the unknowns are unknown. Some of the unknowns are known. So what do we know and what do we don't know, and
is to
what have we not
multiply
yet imagined?
substitute methods to show they are complementary, and that they can, by adding their results together, converge towards the complete substitution of the vital functions of a living animal.
In Europe, progressive adoption of alternative methods and the putting in place of a restrictive judicial framework is leading little by little to a decrease in animal experimentation.
Dans l'utilisation des animaux de laboratoire, les choses...
In the use of laboratory animals, things have changed considerably over the last 30 years. And the installation of European regulations in 1986 has upset mindsets in many ways. It has brought attention to the respect of animals, imposed structure on experimentation, which by increasing prices have driven people to avoid using animals in any old way. At
We
the
see a
same
lot of
time,
patients come
the
into our
advance
coronary
of new
care unit with
methodologies,
acute
new
heart
techniques,
attacks,
have
severe
allowed
heart
all these
attacks,
alternative
life-threatening
methods
heart
to
attacks.
emerge that were
And
extremely
actually, if
rare
you look
30 years
at
ago.
what they're doing in the hours before they come
And
in
which
to
have grown
have
in
their
number
heart
significantly
attacks,
today.
they're 2 to 3 times more likely to
Il
be
y a
in traffic.
une
30 years ago, honestly, in experimentation, anyone at all could do anything at all. And it wasn't because there was no regulation.
That was a good question, actually. We were
Half the
basically driving
experimenters
home
didn't
and,
even realise there was
caught in traffic,
regulation.
and somebody started getting some chest pain.
And the arrival of the European Directive of 86-87 led to scientific circles paying much more attention to animals. And thanks to that, the number of animals used has considerably dropped by 40% between 1990
What we now understand is that there's very small particles that are in the diesel exhaust of traffic. These nanoparticles, we call them combustion-derived nanoparticles because it's when the engine burns them.
Ethical rules
So
and
when people
manipulation
breathe
techniques
in
are now the
these
object of
combustion-derived
special training.
nanoparticulates in the air, they
attention.
can influence the lung, cause inflammation in the lung, and that then causes a cascade of effects that can affect the heart and trigger the heart attack. We also believe that these nanoparticles are so small that they can actually fly across the blood vessel wall, because in the lungs, the blood vessel wall is incredibly thin. So we think these nanoparticles, when they're in the bloodstream, can actually trigger these plaques, this fatty deposit in the artery, to burst open, and that can cause a heart attack to occur.
Is that they will be first and foremost animal lovers, so as to treat them better, to be able to better evaluate the pain involved in the experiment.
My name's Dr. Barnes, I'm one of the researchers involved in the study. Just before we take you out on the walk, we need to put an ECG machine onto you.
Of course, you put yourself in the place of the animal, and you tell yourself that no one likes it when people mess up and hurt us. So therefore, we do everything to make sure that it's done quickly and efficiently.
Are you feeling okay?
Yeah.
In the first months of their training, there is sometimes a bit of reticence, a little fear, which is perfectly normal. Rats, for example, aren't easy animals. But you see very quickly that by teaching them the right gestures and at the same time giving them the philosophy of respect for the animal and of well-being, giving them a lot of advice, you can see that these young people are highly
motivated. It'll be interesting to see what count they get up to. We're about 30,000 at the moment.
The restraint is good. On the other hand,
yeah, and you can see it's jumped up quite a bit there. So it jumped up to about 48,000. Are you feeling okay, Monitor?
Yeah, no, it's fine. Inside a car, the pollution levels are much, much higher than outside the car. They're about 4 or 5 times higher inside a car. So there are 4 or 5 times as nanoparticles that are circulating.
That's caressing, as the English say. Up and down quietly, you see. It's like making bread. Away we go. Now, what I'm giving you here are empty gel capsules. There's no active ingredient, of course. It's simulated so you can learn the movement. Maybe 2 each. That might not be a bad idea. Hey, it's not worth asking for the animal's permission too much.
One of our favourite pubs.
On ouvre par le dessus. Voilà.
It's a nice tour,
so
isn't
open them from above there. No, with the other hand. Are you left-handed?
No, right-handed.
Okay, well, right hand here. Hand here. Like this. Fold back the skin, inside, right inside. Let go now, and you can easily put the gel capsule in.
Et tu peux mettre la gélule au fond. Vous avez réussi ou pas?
Did you manage it or not?
Ah, voilà.
Ah, look at Doggy. Look at those cheeky things. There, you're a little clown. That means you haven't sufficiently, you see, you haven't gone deep enough. Go right down.
Thinking about nanotechnology for me, and I guess many of my colleagues, is a 24-hour-a-day exercise. We can never get it out of our minds. We're thinking all the time about potential applications of the material we're working with, or if we're seeing problems, we're asking how can nanotechnology solve those problems? What new property can we endow in a particle which will solve the problem we're seeing?
The manipulations that I fear the most are, honestly, with dogs, because in the end, they're animals which are really close to us.
The fuel on this bus has got cerium oxide particles suspended in the diesel fuel, and the idea started because of a chance meeting in a pub between someone who had this vision of using cerium oxide to improve diesel engines, and a former student of mine. Clean emission, no soot coming out of the tailpipe, and we see this as a radical way of improving all internal combustion engines and possibly other combustion as well.
Last time they saw how different parts of the isoflurane machine work. They saw the principles by which it functions, the points where you need to check, So next, today it's really the first session where they'll learn, after a demonstration, how to anesthetize the animal. Remember this now, if it knocks out little by little the paws,
if you envisage breaking the cerium oxide down from big lumps down into lots of tiny little nanoparticles, you end up with a very high surface area. And it's that surface area which drives the catalysis and makes it so effective as a diesel fuel
used in experimentation. Rabbits get stressed easily, so well, we're going underneath the chin and then an abdominal massage in order to, of course, relax the animal. Always the way we act towards most species.
More about the environment that exists, but we're also beginning to understand how we can adapt nanotechnology to clean up some of the pretty dreadful contaminants we've introduced there already. I'm one of the optimists. I think that nanotechnology is going to play a much bigger role in improving the environment.
You've got 5 to 10 injections to make on the animal. So there's no special need for restraints. The animal's not suffering. It's a very soft kind of injection. We're not asking you to undress the rabbit. Now you're going to prepare your syringes.
We're beginning to understand
and
more
make 10 injections
about
over a surface of about 10
behaving in the
centimeters.
environment.
You have to know how to
But
make
they're very
an
complex.
intradermal
That behavior is
injection.
complex.
It's a very simple thing,
Technology and
as you can
innovation,
see, and the
like
main discipline
nanotechnologies,
which
doesn't happen
uses
in a
these
vacuum.
injections is immunology.
It happens because it interacts with us as human beings. And the key thing is that we understand that we recognise that we all have our faults, and that technologies can sometimes go wrong. They needn't go wrong if we develop them responsibly. And that's the sheer challenge for us now, with this, probably the most exciting technology of the 21st century.
Responsables de leurs actes.
Regular contact with the animals is essential.
There have been no
we can say that
reported
field training is our strong point, and I think that being constantly present with the animals for
no
basic care and
real
other things
impact
is really necessary.
In fact, we
and this is
haven't
both
really
the application
detected
of the
many
lessons, but also the learning of the
nanoparticles
craft.
in the environment themselves. So, from my own personal
give them a good stroke. That's why we're here.
Not that we've shown that
Go on, you have to caress
they have pose risks.
This is very
it's
important for
simply we don't
laboratory
know.
animals, which are in a box.
Some of the most recent calculations have been
sometimes they need
that
contact.
just the safety testing for those nanoparticles that are manufactured in the US alone, those ones that are on the market now,
Caressing animals, being with them,
30 and 50
it's a way of
years,
compensating
and up
them
to
for
$1
the
billion
difficulties faced by these animals when they're being
It's hard to
studied.
generalise about how long
happy
it's
to
going to
see
take for
people.
us to
Socialising
figure out
dogs,
whether
this is our
nanomaterials
professional ethic.
pose a risk
But
to
it's
the
above all
environment
made
and
compulsory by European and national regulations.
But it's going to take decades, guaranteed. It'll be decades before we fully understand, just for the ones that are out there now, and be able to make decisions on that basis.
It's clear that using alternative methods is a great step forward. Now only a few alternative tests have been validated to replace animal experimentation. It's now an obligation to use alternative testing when it has been validated in relation to
Magda, what's the ORP?
We can say that these are complementary
Well, it should be coming any time now. It should come right through. We haven't seen it yet, and I haven't seen it come through here, but it'll be there very soon. It's going to turn black, I'd say, in the next 40 minutes. So that indicates that we've got good steering of this zone for the remediation of the site.
Thus, in
we're anticipating a widespread acceptance of this technology because we're able to inject the nanoparticles where the contaminants are. A lot of other remediation technologies, you have to dig up an entire site, which is very costly. Then you have to do something with the contaminated soil. So here, this is an in-situ technique right in the ground. We can inject them where they can degrade. We don't have to disrupt the whole site.
Such research can be made partly on isolated cells and on animals like the pig, physiologically close to humans. But they take time, are costly, and only give partial results. Today, mathematical modeling and computer simulation are opening up new perspectives.
In this test, the arsenic in the water is converted into arsine gas, and that gas reacts with the test strips on the top of these vials. And so these test strips change color depending on the amount of concentration in the water.
Jean-Frédéric Jabot is a researcher at the
It's
National
kind of like a
Institute
pH
for
test strip
Research in
on
Computer
steroids.
Science and Control.
How's it going?
Oh, hey.
His challenge consists of
I just,
trying to
just
translate
got
the way our
done with
organs
the
function
reaction
into
period for the
mathematical
arsenic,
formula.
so I'm just checking out our results.
Oh, great, great.
His current research is more specifically on the heart, the arteries, and blood circulation.
That's the before.
That's the before, yeah.
We do the after. After our nano magnetite. Oh, great. Yeah, it's all good.
Yeah, great.
Uh-huh.
Yeah, this is
Right.
zero.
Yeah.
So right now, there is no arsenic coming out of the column. It's being completely
Fabulous. It's called an experiment, but fabulous. It's a really good outcome. So now we just have to keep watching it.
And here the behavior of
I
the
think
cardiac
that
muscle
at
or
this
an
moment in
artery wall.
time,
In this
we are
simulation,
poised,
we've
this is our
represented blood
technology
flow in
developed.
the
This
left
will
ventricle
be
of the
our
heart,
generation's
which arrives here through the
contribution
mitral
to the
valves
world
and
to
is
take it up
ejected
that
into the
next
rest
step.
of the circulation
And so yes,
through
I think my
the
kids are going to
aortic
live a
valves.
better life because of the kinds of nanotechnology that we have where we
Les
can
recherches
put it
qu'on fait
in
permettent-
and solve the
permettent ou
problems
The
that all
research
of
we're
us are
doing
facing.
allows or will allow us to reduce animal experimentation because through digital simulation you can with great ease examine a large number of cases by varying the problem's parameters. And straight away you need less experiments on animals because many scenarios have already been envisaged using the machine.
One of the elements of our work is trying to imagine if you had a nanoparticle in front of you,
which are too
what
dilated,
would
and
it
we're
look
working with a
like
doctor
if
who
it
has
were
dreamed up a
dangerous?
device for reducing the diameter
Would
of
it be
the
blue?
arteries.
Would it be long?
Here you have
What would be the
cases of 4
characteristics of
different
a
patients.
dangerous
Obviously, this
nanomaterial?
device
We don't
will
even
be
know
tested
that
on
yet.
animals
I would be
before it is
surprised if there
implanted
wouldn't
humans.
be some unintended consequence
here
that's
is to
going to get
model
past
and
our
then perform
experimental
experiments on the computer
network.
before making experiments on animals.
We can't anticipate everything, but we can reduce the uncertainty and we can reduce the impact.
Well, the people that lived and worked
all the
in this building behind me have long gone. They lived in a very different society 80 years ago, only 80 years ago to the one that we live in
that's
now.
still science fiction.
And that
It's
society
more than
has been
just to be
shaped
looked
by
forward to, it's still
successive
science
waves
fiction.
of technology and innovation. Nuclear technology, molecular biology, information technology, and now nanotechnology. And my grandchildren will live in a very different world again, shaped again by innovation and technology. And as human beings, that's what makes us, our creativity and our appetite to move on and be innovative. But we also know that we have to be responsible. With an exciting technology like nanotechnology, we don't want to be left with a legacy for our grandchildren to have to pick up. So a lot of the questions that we have about new technologies are not just technical risk questions, they're philosophical questions. What kind of technology do we want? Is it safe? Should people have a say in shaping technology? Do we live better lives? Does it have unintended consequences? Does it create inequalities? We accept these technologies, we buy these goods, we move on. What we should probably do is be a little bit more reflexive, think a little bit more about those technologies and about the world in which they are deployed and how we live with those technologies in that world.